Seconds. There it goes. Now it's recording. Maybe not. Yeah, it is recording. It's got seconds right there. Okay. So this is indeed pseudostratified columnar epithelium with cilia and goblet cells. And then we do have, whoa, here's the nice little cilia buddies there. They are connected with a kinetochore. So there's a little protein connection for all of these little hairy guys. So that's a dead giveaway that this is not microvilli, that it's actually going to be the cilia. Okay? So we always look for that stain right at the edge there. These guys are all individually, even though they look like they have their nuclei at different levels. It appears stratified, it's not stratified. Every single cell actually touches the basement membrane. So that's an important detail of this. This is also going to be our standard respiratory epithelium. So people who are put upon for various reasons, whether it's through smoking cigarettes or whether it's through working in a coal mine, and there's still a lot of people working in coal mines, that means that this tissue is going to be challenged, and very often it will make an adjustment to this tissue, which is the esophagus. Let's get our tissues in here. Here's the esophagus now. The esophagus is right next to the trachea. In fact, it's not next to laterally, but it's next to in terms of posterior to the trachea. So that means that these tissues form together, they differentiate together, they're activated together, and so consequently, when one tissue decides to go to a different tissue type through transformation, the beginning of malignancy, then this is the selection that's often made, and we have squamous cell carcinoma. Okay, so this is going to be the stratified squamous. It's not outside the body, it's inside the body. So the oral cavity and the esophagus will look like this. And we're going to have the basal cells here, very dark staining, which means that they're going to be engaged in mitotic division. And so occasionally, if we were to get somehow or another close enough, but I don't have enough ability. If I went to high density, I might be able to see some of those mitotic figures, but I don't. The surface area cells up here at the very top are going to be squamous. The characteristic of these tissues is always at the apical surface, away from the basilar surface. So we can tell this is not a transitional epithelium, that it's a squamous cell. A stratified squamous epithelium. It's kind of nice to go down here and to actually see some of the connective tissues that underlie this tissue. So we have a combination of dense regular and dense irregular connective tissues, or rather dense irregular and areolar, dense irregular and areolar. And we actually get to see some skeletal muscle right here. Okay, because that's what we use for swallowing. In the meantime, nearby, not too far away, in the trachea, we're going to have this nice cartilage, which is of the hyaline cartilage persuasion, and that means that inside of each of these lacuna is going to be living a chondrocyte. Some of these are present, some not present. And we have, remember that the types of matrix here involved are ground substance that is no longer fluid, it becomes solid, and there's going to be more or less invisible or slightly visible little areas of collagen fiber. Okay, we'll call it quits on